Okay, this is a review for exam 3, which will be on Monday, November 21st. Um, Remember, kind of the same study tips as always. Spend lots of time. Repetition is the best way to learn to remember things. And in particular, choose active methods where you're actually writing notes or review sheeting or actively quizzing or being quizzed. Um, it's best to do these things over several days rather than one day, and to space them out so that you can learn. So we're going to cover a couple different chapters here with a bunch of slides. Chapter 12 was started out with talking about the divisions of the nervous system. Uh, the nervous system can be divided into the central nervous system, which is the brain and spinal cord, and those structures, and then the peripheral nervous system, which are the nerves and ganglia outside the central nervous system, and that includes the spinal nerves and the cranial nerves. Okay, we also can talk about the autonomic and the somatic nervous system and various divisions, but this is the important structural division. Here is also where they introduce the different cell types in the nervous system. The neurons are the most crucial cells. Neurons are large, long-lived cells with extensions. The important thing about neurons is that they can receive stimuli and transmit action potentials. Their body has a large cell body or soma. Their receptive surfaces that receive messages are dendrites, and the surfaces or extensions that transmit messages is actually just one. It's the axon, and that leads to other cells. Neurons, we talked about the function of neurons in this chapter, and one of the important things is to know that the neurons maintain an electric potential across their cell membrane. This the resting potential is a gradient in charge and in ion concentration. There are more positive ions outside than inside, so the neuron generally has a negative charge at rest, and this is due to the distribution of sodium ions. There are many more sodium ions outside the cell than inside the cell, and this is maintained by a pump and then some leak channels. Neurons have the potential to undergo an action potential or depolarization. And when a change in charge hits a threshold, um, that leads to a opening of ion channels. That leads to a rapid depolarization of the cell, where the cell actually becomes positive on the inside. Okay, and then that potential is quickly reversed to restore an resting potential, and so that happens when other ion channels open and allow potassium to diffuse out. So the action potential, the rising phase is due to the influx of sodium. The falling phase is due to the outflow of potassium ions. Okay, and this is due to voltage sensitive ion channels. Okay, neuron membrane potentials can lead to signaling of other cells at a synapse, a junction between cells. Most synapses are chemical synapses, where the action potential of the presynaptic cell leads to vesicles releasing neurotransmitter at the junction with a postsynaptic cell. And those neurotransmitters bind to receptor molecules and cause changes in the postsynaptic cell. If they're excitatory, usually these changes are to open sodium channels, allowing the membrane to become depolarized, and so that starts an action potential. But they could also be inhibitory. Some neurotransmitters will cause the opening of potassium channels, making the neuron more negative. All right, so that was probably most of chapter 12. There was a discussion of things like the neuroglial cells. Most important among those are the myelinating cells, like oligodendrocytes or Schwann cells that speed action potentials. But then we started chapter 13, which was over the structure of the nervous system. And so it began with the structure of the brain. We talked about the structure of the cerebrum, which is divided into left and right hemispheres. The outer layer of gray matter forms the cortex. There are frontal, parietal, temporal, and occipital lobes. Those lobes have distinct functions. Underneath the cerebrum, it's connected to the rest of the brain by the diencephalon. The diencephalon has two major structures, the thalamus and the hypothalamus. The thalamus serves as a sensory relay. Most sensory information that comes to the cerebrum comes through the thalamus. The only one that doesn't is the sense of smell, olfaction. The hypothalamus is an important autonomic center. It is where the homeostatic control center of most functions occur. There's a gland that's part of the diencephalon, the pineal gland, important in wake and sleep cycles. Then posterior to the diencephalon is the brainstem, the midbrain, the pons, and the medulla oblongata. These are autonomic areas that control many other functions. We're not consciously aware of this. These include reflexes, like in the eye positions and things like that, auditory reflexes, visual reflexes, breathing, circulatory system things. Those are controlled in the brainstem, medulla largely, but also pons and midbrain. And then just located posterior to the brain stem is the cerebellum. It too has an outer layer of gray matter. It's involved in motor control. It gets a copy of information from the frontal lobe, and it compares that to incoming sensory data and helps carry out things like posture and complex motor movement. And it helps us coordinate those things without having to think about it all the time in the cerebrum. Okay, when we talk about the cerebrum, we can also talk about systems that are connected between these different regions, things like the limbic system, which includes parts of the cerebral cortex, the thalamus, the hypothalamus, and other brain regions. This is involved in memory, reproduction, nutrition, emotional interpretation of sensory input and emotions in general. We also talked about the reticular formation, which controls awareness and activity of higher centers, involves many of the regions of the brainstem. In the central nervous system, in addition to these structures in the brain, there's the spinal cord. The spinal cord goes through the vertebral foramen of the vertebrae. It gives off the spinal nerves, which are part of the peripheral nervous system. There's the cross section there in the lower left. It serves as a center for reflexes, and it carries information up and down the spinal cord to the brain. So you have stuff in there. The central canal is continuous with the ventricles of the brain. It's also surrounded by the meninges. Again, giving off 31 pairs of nerves, ending in the cauda equina, a bunch of nerves that continue down in the vertebral foramen, and has enlarged regions where many nerves for the arms and legs come in. Those are the cervical enlargements and then the lumbar enlargements. In the peripheral nervous system, that consists of the 12 pair of cranial nerves and the 31 pair of spinal nerves. Each spinal nerve has a nerve root. The dorsal root is sensory. The ventral root is motor, and those roots combine to form a spinal nerve that's a mixed nerve. Some of those spinal nerves will be mostly somatic, going to voluntary areas of the brain. Others are going to autonomic, to visceral organs, to things like blood vessels and organs like that. The 12 pair of cranial nerves that are numbered 1 through 12. These include the optic, the olfactory, optic, ocular, motor, trochlear, trigeminal, abducens, facial, vestibular, cochlear, etc., etc., etc. These nerves, some of these are going to big sensory organs like the olfactory nerve, the optic nerve, the vestibular cochlear nerve. Others are entirely motor, like the ocular motor, trochlear, the abducens, and then many are mixed, like the facial nerve. And so those nerves, most of the going to structures in the head and face and neck, particularly the throat, but some to also visceral organs. Nerves themselves really contain the axons and dendrites of nerve cells and not the cell bodies. And these individual axons or dendrites are surrounded by a small layer of connective tissue called the endoneurium. Groups of these things are bundled together in fascicles, also surrounded by connective tissue. And then a bunch of fascicles are surrounded by an epineurium. Okay, and these will go to different parts of the body. Some will carry sensory information, others are going to carry motor. The peripheral nervous system also contains some ganglia, which are collections of nerve cell bodies outside the central nervous system. And here's where there might be a little bit of processing, but most of it's just reflex activity. Okay, in the peripheral nervous system, you have these 31 pair of spinal nerves. Those are coming out between the vertebrae at the intervertebral foramen. Spinal nerves have cutaneous distributions called dermatomes, but they also have branches going to visceral organs. They combine and recombine, especially in areas where there's the arms and legs come in. These leads to plexus, the cervical plexus and the brachial plexus, things like that. The nerves carry information. Most of that information is trapping rapidly because it's going very, very quickly. And it needs to go quickly for reflexes. And so, like in this case, this is the stretch reflex where a reflex hammer is used to hit the patellar ligament, and that causes a stretch receptor in the muscle to become active, and that will send an impulse to the spinal cord where the reflex is centered, and then a motor nerve will cause the muscle to contract. It allows us to respond quickly to a stimulus so that we can maintain balance. Okay, I mentioned the cranial nerves. They're listed there. 12 pair, and you can see them there. Those are numbered 1 through 12 for whether they come out anterior, like the olfactory and optic, or posterior, like the accessory and hypoglossal. Okay. Then we switch to chapter 14. Yeah, chapter 14, which deals with the senses. And we start by talking about the stretch receptors and things like simple receptor organs where it's just a modified nerve cell. And the general senses are generally like that. And those senses are distributed throughout the skin, but particularly in areas like the fingers and face and the genital regions. And those project to the parietal lobe of the brain. The special senses usually are senses where the information is carried back to the brain by cranial nerves, and there are large, complex organs involved. And in particular, we spent a lot of time with the inner ear, where we detect hearing and equilibrium. Here's a structure of the 3 layers of the ear: the external, middle ear, and inner ear. And so sound is conducted to the tympanic membrane, which vibrates and causes the vibration of bones. And those vibrations that are from the bones are carried into fluid of the cochlea. And in the cochlea, that fluid vibrates in a fluid-filled canal called the organ of Corti. And the receptor cells in there are referred to as hair cells, and they're mechanoreceptors. And they have these stereocilia on their surface, and when those brush against the tectoral membrane, that sends a message back to the brain, and depending where those hair cells are, the brain detects that as sound of a certain wavelength. We have maybe 10,000 of those receptors in our cochlea. Okay, the inner ear also contains the structures of the vestibule, the utricle and saccula, and these detect head position, part of our equilibrium sense. And there you have otoliths embedded in a gel, and gravity causes that gel to slump at certain times, and that triggers the receptors. And depending on which receptors are triggered, that allows us to detect the position of our head, part of our sense of balance. Okay. The other part of this sense occurs in the semicircular canals, which detect rotation in a particular plane. And here we have, in a little expanded area at the base of these canals, an ampulla, where there's, again, hair cells embedded in a gel. And when the fluid flows past that cupula, it causes it to bend, and the hair cells detect that and send a message back by the vestibular nerve, cranial nerve 8. And so that we detect that as a rotation. Okay, so we talked a little bit about pathways with this. The pathway for sound is that cochlear nerve, part of cranial nerve 8, goes to the cochlear nucleus, and then the part of the midbrain called the inferior colliculi, and then through the thalamus, where it's evaluated, and up to the auditory cortex. All right. Okay, vision. Vision is detected through the eye, which has, again, 3 layers. The outer layer includes the cornea and the sclera. It's made of collagen, modified in the anterior end to be clear. This is the tunic, the fibrous tunic. And then inside of that, we wind up with these cavities and a bunch of structures. This is part of the vascular tunic. The first chamber is the anterior chamber, which contains aqueous humor. Okay, then posterior to that is the iris, a circular muscle that has an opening, the pupil that lets light into the eye. The lens, along with the cornea, focus light on the retina. It's held in position by ligaments, and its shape can be changed by a muscle called the ciliary body. Okay. Then posterior to the ciliary body, there's a layer of pigmented cells called the choroid. They absorb light that passes through the retina. The retina is the part of the nervous tunic that detects light. It contains the receptor cells. Okay. Those cells send a message back by the optic nerve. Okay. Here, when we talk about eye physiology, hold on, let's go back. I got lost here. Hold on. The eye physiology includes the physiology of the receptor cells, and then the light and dark adaptation. The receptor cells are not all the same. There are more receptor cells in the center part of the retina. That's the fovea. There's a place called the blind spot where the optic nerve and blood vessels enter the eye, where there's actually no receptor cells. Okay, the receptor cells in the fovea are actually color receptors, so they're slightly different type. These are the cones, and the cones respond to 3 different colors, and each cone sends a direct message to the brain by way of its own sensory neuron. The other type of receptor cells are rods. Rods are more sensitive, and several rods will work together to send a message back to the brain. And so when we want to see something really precisely, we have to look at it in an area where there's lots of cones, because rods are good at detecting light, but not so good at localizing it. All right. Adjustment to light and darkness. That happens by way of the iris, which can open and close the pupil. That's a quick response, but it also turns out that these cells, the rods and cones, can accumulate more pigment, the photoreceptive pigment. And so over a period of 10 to 15 minutes, they become more and more sensitive. All right, so all this stuff projects back to the brain via cranial nerves. Okay, everything is going to be first evaluated in the thalamus except for the sense of smell. And then we have specific areas of the brain that deal with that. The primary somatic sensory cortexes deal with our sense of touch. We have sensory areas for taste in the temporal lobe, sensory or parietal lobe, auditory areas in the temporal lobe, speech areas in the temporal lobe, and then visual areas in the occipital cortex. The last thing we have to talk about is motor control. The somatic system, voluntary interpretation and voluntary perception occur in those three posterior lobes. Motor control occurs in the anterior frontal lobe. And we've got localized things that we can say for the eye movements, for the movements associated with the production of speech, Broca's area, and then the voluntary muscles throughout the body. That's the primary motor cortex. We have planning areas for movement, include the premotor area and prefrontal areas. Prefrontal areas are also where we have executive functions like memory, judgment, things like that. So we'll talk about those more in chapter 16. Motor control is a pretty integrated function that the cortex and the frontal and prefrontal areas are involved in planning and learning of motor areas. And then it gets pretty complex. We have basal ganglia, the cerebellum involved, and other kind of areas like the midbrain. And these areas are eventually all going to send information via the spinal cord to the voluntary muscles. There's two main pathways we have to kind of know about. Those include the direct pathways, the cortico-lobar and corticospinal. These go through the midbrain and are going fairly directly to the lower motor nerves that are in the spinal cord. So there's a synapse with those. We use these often when we're learning new skills, and so very important that way. Then we have lots of indirect pathways, and these indirect pathways involve communication between several brain structures. The cerebrum decides on what's the intended movement and send messages often to the red nucleus, which will relay information to and from the cerebellum, which compares intended movements from the cerebrum with incoming sensory information and sends material back to the red nucleus and then down these extrapyramidal tracts that are some of these are related for posture, like the cerebrospinal, and others are going to the voluntary muscles. I probably won't ask you about any of these tracks directly, but they're fairly complex. They cross over often in the spinal cord, and they're sending information to muscles of the trunk and limbs. They're also, when we talk about those things, it's kind of important to talk about how complex this is. And so I talked about speech and the coordination between different brain areas that are needed to produce the voluntary activity of speech. And so the main areas are Broca's areas and Wernicke's areas, but this also can involve occipital areas, temporal areas, parietal areas, and things like that. Okay, so I wanted to say a few things about the essay questions. And so with the essay questions, the first question is, what is a neurotransmitter? How does it work? How does a neurotransmitter produce an effect at a postsynaptic cell? Why are neurotransmitters important? And support your answer using specific examples. And so neurotransmitters are covered in chapter 12, particularly in section 12.5, where it talks about synapses. Okay, and so And it talks about different neurotransmitter systems. It goes through examples like acetylcholine, the amino acids glutamate, glycine, and GABA, and then the amines, serine, dopamine, norepinephrine, epinephrine. So there's lots of examples in chapter 12. Briefly, all neurotransmitters are released from vesicles that are stored in the synaptic bulb. Okay, that happens after an action potential moves down and causes the release of calcium into the neuron. The neurotransmitters diffuse across the gap. They all act by binding to receptors. Usually, those receptors are ion channels that will open. If it's a sodium channel, it will be excitatory. If it's a chloride channel or a channel for potassium, they will be inhibitory. Okay, a good example is acetylcholine. It has two different types of receptors. The nicotinic receptors are usually excitatory. The muscarinic receptors are usually inhibitory. But there's many, many examples. And so that's really chapter 5, section 12.5. Look in there, read that over. There's also on one of the PowerPoints, there's a couple slides that go through some examples. All right, the second example here is describe this, or essay question is describe the structure and function of the diencephalon. What brain regions are found there and what other brain regions have connections to the diencephalon? Can you describe diencephalon functions? Okay, so this is chapter 13, and section 13.2 is entitled diencephalon. The diencephalon sits below the cerebrum. All information coming to the cerebrum has to go through the diencephalon. It also forms the walls of the third ventricle and connects to the brainstem. The thalamus is in the diencephalon. The hypothalamus is there. There's a choroid plexus attached there. The thalamus. Okay, it's an important sensory relay. That means there's some processing of sensory information there. Everything except olfaction goes through the thalamus. There's also motor connections have to pass through this region. The hypothalamus is a really important center for autonomic nervous activity. It's involved in homeostasis. It's important in memory and emotion. These are part of that limbic system. And so that look at that. There's some information there on chapter 14 on the diencephalon, but most of that's chapter 13. Okay, then all right. So the third question: describe the sense of equilibrium that depends on our inner ear. What inner ear structures are involved? How do these structures respond to head position or movement? What brain structure handles this information? What else contributes to our sense of balance? Okay, so these inner ear structures. This is part of section 14.1 on sensory perception. It's the section on equilibrium. Okay, the vestibule is one of the structures that contains the utricle and saccule, and inside of those chambers you have the maculae. That's it's got a jelly-like membrane that has stones embedded in, and gravity causes that to slump depending upon our head position. The receptor cells in there are mechanoreceptors, and we're going to we're going to interpret that as our head is in this position. The other big structure involved are the semicircular canals. These are fluid-filled rings that have a little expanded ampulla at their base with, again, mechanoreceptors embedded in gel. And when the fluid flows past there, we interpret that as rotation. So all this information goes back by cranial nerve 8, the vestibular cochlear nerve. It's going to nuclei in the medulla, the brainstem. It's communicating with the cerebellum and the cortex, and that's how we determine, part of the way we determine equilibrium. But there's other information. Stretch receptors that are sending information back are involved in balance. Our visual information is involved in balance. And so there's a lot more than just that. Okay. All All right, so I'm going to stop there. Plenty of stuff to do there. I will be in my office before the exam. Again, the exam Monday, November 21st, and hope to see you there and hope you get ready.